And of course we've had a very wet week, and that's just what these plants love, because it makes mud. And these plants grow in mud. Previously I've spoken about very colourful plants, but and these ones are not without their colour, they're noted for being red, but I felt that it would be useful to talk about a group that many people won't be aware of, and yet it's a real botanical treasure, particularly for Western Australia, of course, for Western Australia. So we'll get going. Just a little acknowledgement for some people. I've had quite a number of collaborations with people from various places in Australia and overseas, and you'll see that that's the case. And Dmitry Sokolov is probably the major one that I've worked with a lot, who was in Moscow, he's now in Tel Aviv for good reasons. And then there's collectors without whom we just wouldn't know where these plants are growing. Often found kind of incidentally, and over the years these people have taken a little bit of an interest or just happened to come across them, and that's built up our database and knowledge of these things. So just thought I'd start by explaining about the names. So the genus Trithuria was named by Hooker in 1858 in this work here, for Tasmania, and it means three holes or windows, and it refers to the way that the fruits open. They look like three little doors. In those that, yes, the first one that he came across happened to do that. And then there's a synonym, Hydrotella, which we won't hear too much more about because it's now they've all been put into Trithuria. But the meaning of that was not given in the original publication, and so there's a couple of variations of the interpretation of it, but I've just simplified it to referring to the habitat growing in water. And the Trithuria is the only genus in the family Australasian. And the reason why it's called Australasian and not Trithuriaceae, which is the older name, is because it was, the person who created the family Australasian, Harman, happened to be working on one that was classified in Hydrotella, I guess. So that was the thing he knew, and so he used that as the foundation of the family. But now we're left with a family that uses the name of a genus that's not current, but that's not uncommon. I should also mention that we just have to get used to calling them Trithuria. There are no common names for these plants. I've only come across one, and that was just the one created for a threatened species, and that was referred to Hydrotella. It isn't probably appropriate anyhow. So, and also I don't know of any indigenous names. And so these plants are they're rather obscure, and they don't have any economic value that I know of, except bringing scientists to Australia to spend their money. But of course they must have an important role in ecology. Everything's intertwined. Some habitats where you find these things. This is a seasonal clay-based wetland with a dominance of Melaleuca lateritia there. So that would dry out over summer, but probably could have standing water in the winter time. That's a common kind of habitat. Standing water, again a clay-based wetland, but without that Melaleuca in this case. This is from near Manjimup, and then that will dry out completely as well. And there's lots and lots of the plants in good season. Lots and lots of Trithuria growing in July over the surface of this thing. And pools on rocks is a common place, especially along the south coast. That's from the Ravensthorpe area. And then moist sand is another possibility, usually near a wetland. That's the smallest of our Trithurias. That's Trithuria bibracteata, which doesn't turn up much more in this talk. So Trithuria plant structure, they're nearly all annuals, and they're herbs with almost no fibre in the leaves. The leaves just die off at the end of the season and vanish. So this is an inflorescence, and they're usually on a peduncle like that, although bibracteata is very sessile, really, hidden a bit down among the leaves. So this has got a couple of bracts around it. This is Trithuria austinensis, just escaping for the moment, named after Austin Bay Nature Reserve near Major. But it's much more widespread than that. It ends all the way to Cape Arab. So inside the bracts there are these flowers, several flowers or many flowers, and the flowers are unisexual, so they're just they're male or they're female, and they can be mixed or solitary, single sex, and we'll get to that in a moment. But anyway, basically the flowers are extremely simple. The male flower is just a stamen, and the female flower is just a stalked ovary, but growing out of the chips is a whole tuft of hairs. It's not a proper stigma, they're called stigmatic hairs, and you can see some here. Oops. Yeah, those are stigmatic hairs. This photo was taken on a sheet of plastic with a bit of water lying around it, so it's skew a bit. And here you've got more than one stamen showing more than one flower. So here we've got this explains the sex situation of these things. Trithuria submersa has a mix of male and female, and usually there are quite a few female flowers and just one or two male flowers consistently. That's a good way to differentiating it for differentiating from another species we'll come to later. Trithuria australis has usually short but can be longer inflorescences like that. And one of the interesting things about this one is it's monoecious. It's got female inflorescences and male inflorescences mixed in here. And usually there are far more female inflorescences than male inflorescences. And then finally we've got austinensis that I referred to, peeking out of water here. So what they've done here is they've been growing underwater for a few months, and as the water level recedes, they start to flower. They're all ready to go, and when the water level drops, they flower. And these are female plants here and male plants here. So I wanted to tell some stories about some interesting research projects that we did. So this one is about the Trithuria australis group. So Australis was named in about 1904, and it became apparent in our work that it was rather variable, and also there was some genetic data that showed they weren't all the same thing. So we did a systematic systematic study, collecting them from throughout the range as far as we could, and then studying them to see if we could find morphological differences that would enable us to tell apart those genetic groups. So this diagram here is the result of it's a mapping of the morphology and the genetics. So down here it says that plastic phylogeny. So those shapes refer to the groupings found in the DNA study. So there's 4 groups shown there. Group 1 is here, and actually it isn't really there at that site. That was while we were writing the paper, we thought we had worked out where that sample came from. I'm afraid we were a bit careless at one point when collecting a sample, and I had collected some seeds, at least I believe I collected some seeds with someone else, and the seeds, some of the seeds were sent to the Royal Botanic Gardens queue where they grew them, and they had to work out how to do that. And then they got some DNA samples from that. And it is the only member of its group. But the fact is that we don't actually know where it was collected. I think it was collected from near the Unicup area here, but we don't know for sure. So that one we can disregard. There are 2 major groups, the ones indicated by the squares and the ones indicated by the circles. And this is a tricky situation here because we don't know which is the true Australis. The type comes from here on the south coast of the Hamersley River, and named by Deals in 1904, the first time the species has been collected, I think. And then he also collected another one here at the Hutt River. And try as we might, they've never been found in either of those places since. And we think it's probably because of the changing water quality, salinisation of the rivers. But I haven't given up. I've spent a fair bit of time in both those places and I'm not going anymore. So we don't have DNA samples for either of those, but we do have hopes of getting some from the type specimen, which the main type is in the Berlin Herbarium, where we have a small amount in Perth. And Berlin have agreed to let us sample that for the study we're doing, because you can use quite small ones these days. Now, in this group here, that's group 3, there's another group 4, which is, no, hang on, it's 2, sorry, group 2, which is recognisable in the DNA analysis, but not very easily morphologically, and it all comes from one place. We've got several samples of it. It all comes from one wetland, which is an outline one here. And so then we need to find out the differentiating features. So my Russian colleagues who did these SEMs noticed that these ones here, which became the new species Fitzgeraldii, have these papillae on the fruits. And these ones, which we call Australis, we think that's probably the type form, because we've been able to look at the fruits of the type specimen, and they agree with that. They are smooth. They've got a smooth coat, not one of these papillae. Generally smooth pericarp, but it's got little granules all over it. And we've looked at multiple samples, and we know it's not dust, which was the first query. And so there is a morphological feature there, and it could be that one of these days we'll bite the bullet and this one here is a light microscope image. So you might take fright and think you've got to have a scanning electron microscope to see this feature, but you can see it under a high-power light microscope, and you can even see it on a dissecting microscope if you know what you're looking for. This is the new species Fitzgeraldi from near Manjimup. So that's what it looks like as the water recedes. These plants are probably finished flowering now, and they're just in the mud. And where the water's deeper, they might still not yet come into flower. So the flowering follows the decline of the water. And that's one single plant there with a whole mass of flowers. These are extraordinarily big plants for Trithurias. It was a very good season that year. A story about Trithuria submersa. So submersa grows across Australia in the southwest, South Australia, southeast, just getting into New South Wales, and in Tasmania. And we did, so that was of interest to see whether it was genetically uniform or not. So DNA analysis is done. Those dots there are the samples, and it turned out that the eastern and southeastern ones, including South Australia, are genetically different from the southwestern ones, and that's another plot to show that. And this is the group of species that submersa belongs to, and you can see here, this is a diagram of the genetic relationships, and here we've got a very short internode there, which shows that they're very close genetically. So that's southwestern Australia, southeastern Australia. So far we don't have any morphological differences between those two areas. We probably can spend some more time looking for that, and maybe one of these days they'll be named as different species. The third story is the Trithuria occidentalis story. So there are plants that look like this, which were collected from the Midland area on the coastal plain. These grow in the seasonal wetlands. In fact, this photo was taken by a colleague, Renee Tuckett, from the Pemberton Nature Reserve, and that's a seasonal clay-based wetland that dries out in summer. And there were the plants looking like this were first collected as far as I know by Alexander Morrison and William Vincent Fitzgerald and a couple of others around that period, 1898 to 1902. There was a bit of a flurry of collecting at that time, and it may be that there was an active botanical group in Perth, and it may be that also there were quite a lot of fires at that time. There's seen evidence from other plant groups. So in the metropolitan area they may have been doing burning, and these things are visible after fires. They were studied at Kew by Otto Stapp, mainly a grass taxonomist, but he looked at other things. He was an Austrian botanist, very well trained, and he found these very interesting, as I'll show you in a moment. And he gave these ones the name Trithuria Macfarlane Trithuria, but he never published it. He did the work, but he didn't write it up for some reason. And the name was seldom used after that, but it was used in the Blackall and Greaves books as an invalid name. But the plant was different, so why not? Actually, they misspelled it as Macfarlane Trithuria, but we'll forget that. The name means large anthers, quite large for this group. And then eventually they were named by David Cook in 1983 as Hydrocella dioica. And he made the comment that female heads have not been seen, and the species is possibly extinct because of clear, development on the coastal plain. Well, then afterwards, Greg Karri found plants like that at Ellenbrook. He was always very interested in these kinds of habitats, and he knew about the Cook's publication. Of course, I think they'd been in touch, and so it was not extinct. But he and others looked for female plants for quite a long time and didn't find them. So this is a sheet, a herbarium sheet, or a piece of paper the size of a herbarium sheet at Kew Herbarium, which has drawings by staff, at least I believe that is. He did this in other groups as well. He would do his technical drawings on a sheet like that and leave it in the herbarium. So he had all the information for describing it, but he didn't do it. So then we come to these plants. Plants like this were first collected by James Drummond about 1840. They were, years later, they were validly named Trithuria occidentalis, the Western Trithuria, by Bentham in Historia Australiensis. And also on that previous picture, Staff must have written back to Fitzgerald, I think, to ask him about what other plants were growing with this thing that wherever he found it. And one of the species mentioned there is Trithuria occidentalis. So there's no specimen as far as I know from that time, but Fitzgerald must have worked that out. And however, that species was then put aside. It became a synonym of Trithuria submersa, which it looks so much like. The only thing is that if you look at these heads here, remember I told you that submersa has one or two stamens sticking out of the heads of the female flowers. This one doesn't have any stamens. So if you get down in the mud and get the leaves all muddy and use a hand lens, you can use that feature to tell them apart. So this is the plant at Anthesis. These are the stigmatic hairs, and this is a bit later when they're starting to set seed. And in a study that Sokolov and others, including me, was involved in revising the taxonomy of this group, we realised it was really Dimitri who realised, because he had access to all this stuff at Kew, that occidentalis was really a different species. It's got, one of the differences is it's got more bracts around the head. It's got about 6 to 8, and submersa nearly always has 4. And the seeds have a different pattern. You can use that to work fairly readily. So this turned out to be the female. Trithuria occidentalis is female, as described by Bentham, and it's the female of that other plant that we saw. So here they are, united at last. Here's the male and there's the female.
Romantic.
Yes. So I wanted to perhaps go a little more into the history about the discovery of Trithuria. They were found in Australia before they were found anywhere else. I think I've forgotten to tell you that Trithuria grows in Australia and New Zealand, and one species in India. And in Australia, there's the one species that grows across Australia, submersa, and one species in Tasmania, but no others in the east, except in the tropics where there's another widespread species, Lanterna goes from Western Australia to Queensland, and a couple of other narrowly distributed ones. I'm afraid I forgot to include a map. I have a map ready. And in southwestern Australia we've got about 8. So there's by far the greatest number in southwestern Australia. So the first description of it was by J.D. Hooker in the book I already showed you. And he based his description of the genus and its first species, Submersa, on plants that were collected by Ronald Gunn in Tasmania and by Muir in southeastern Australia. And Gunn's collection was from the bottom of a lagoon near Macquarie Harbour, and he also referred to the red colouring of the plants. They were quite distinctive and noticeable. And Ferdinand Muir from Melbourne, collected them from Hopkins River and Mount Emu Creek in Victoria. I should have shown you that diagram there. This is from a colour painting from Hooker's book. And it's a marvellous piece of artwork, one of the best illustrations of these plants, and it was all done from dried specimens, which is quite remarkable. These 3 here are Central Epidaceae, and this is Trithuria submersa, and it shows the breaking up of the fruits there that suggested the name to them.
Are those 2 genera related in some way? They're on the same
Centrolepis.
Okay.
We're going to hear more about that.
Because they were starting to look like that to me.
Yeah. So, but the question is, who really collected Trithuria first out of these? It can be tricky to find the dates of these early collections, and so I've looked into it. And Gunn's collection from Macquarie Harbour was made in November 1845. And there was another collector, Charles Stewart, who collected it in 1849. And then Mueller's collections were made from somewhere after 1851, maybe 1852, but we don't really know because it collected there a number of times over the years, but not before 1852. And some of his collections do have dates, so 1853 and 1854. So the winner is not any of those, but James Drummond, who had probably around 1840, could have even been 1839, I'm not sure. But and this is this specimen of Trithuria occidentalis at Kew, that's now regarded as the lecture type, and that doesn't have a date on it. And there are copies of this in about 6 herbaria around the world, including Perth, and only one of them has a date on it, and that's the one in Paris, where they meticulously recorded when they received things, and they received it in 1843. So it was clearly collected before 1843, which means it was earlier than Gunn's. So there you go. It was here where the homeland of Trithuria is. It's collected first. Now, from then on, so obviously around 1840, 1850s, early 1850s, there was a bit of collecting of Trithuria going on, but then it was scarcely collected after that. I haven't really gone into the subsequent history of the eastern states, but in Western Australia, there were no more collections that I'm aware of until 1898, when Morrison collected some, and in that period, Alexander Morrison, the government botanist here at the time, W.B. Fitzgerald, an independent botanist and geologist, and the German visitor Ludwig Diels all collected it, different species of it. And then and actually, I just mentioned that Beals, who we've already heard about, collected the Australis that we can't find anymore in a book with Fritzel. He named Hydrotella, and there are two species, Australis and the one from Hutt River, Letubani. And that's a really very nice illustration of the Hydrotella. Then really there wasn't much collecting that I'm aware of until about this period when Bob Royce from the then of the Herbarium collected a number of times. Royce apparently was, I'm sure Alex knows a lot about this, but Royce was known for collecting small plants. He, is that right, Alex? He called them belly plants or something. He had an eye for small things. So he collected it a number of times. And then there was a collection by Paul Wilson in 1967, and Alex collected in 77, though he was just telling me a little while ago that he hasn't collected it. But we've got proof. And then from 1978, when Greg Curie started collecting, and quite a number of others as well during that period, that Greg has collected consistently for many years. It's been his collecting has been foundational for this group. And then my involvement started but not so serious until several years after that. Now we come to the story of the classification of Trithuria. This is where we get into the part of my title where I talk about the scientific importance of this group. It's quite interesting. So I mentioned that Hooker was the first to describe it, but actually Mueller had, as I mentioned, collected it in Victoria, and he'd studied it, and he'd given it a name. He realised it was something new, and he gave it a name, but it was never validly published, so we don't use that name. And he thought it belonged in the family Centralepidaceae. That's a Centralipus species there, and that's Trithuria australis. When Hooker named, a few years later, named the Trithuria, he said it's a singular little plant of very obscure affinity, but he followed Mueller and placed it with Centralipus. I wouldn't say in it, but he's placed it next to Centralipus in his book. And then later on, another German botanist, Hieronymus, made Trithuria a tribe, or at least he created a tribe in Centralepidaceae for Trithuria because it differed from the rest in having two-celled anthers. If you look very closely, you can see that that one there has got two lobes on it. And this one's split open, but it's side on, so it's an apparent there. Instead of, and also the filaments are attached to the base there, and it's quite a stiff structure. It's not flopping around. Centralipus, on the other hand, has single-celled anthers, and they're versatile. That is, they're just attached by a thread, and they can move around. So that's a major difference, and that aspect, the single-celled anthers, is a feature of the Restinaceae. And Centralepidaceae sometimes these days put into Restinaceae, even though they're from different plants. It's difference between rush-like long-lived perennials and these small, mostly annuals. The structure of the ovaries in these things is different from Trithuria as well, and they kind of stack there Then the Trithuria, seeing these remarkable differences at the tribal level within Canis and Calabidaceae, Harman decided to have a closer look. And I think he was a person who specialised in looking at embryology and fine structure of the ovaries and so on, and reproduction of plants. And he listed a whole set of differences between the central epids and Trithuria and Hypatella. And so he named it Hypatellacea. And this was generally accepted scientifically. I think that I can't any doubts about that. It was taken up. And he stated that the affinities of the Hypatellacea are still obscure. Molecular evidence is highly desirable, which is what we always say these days. And he was writing this later than 1976. It was a book chapter in later years when DNA was becoming available. But it took quite a long time before anyone was able to believe material and analyse the DNA. And one of the people who did was Barbara Briggs from the Royal Botanic Gardens in Sydney, who had access to the molecular lab. And she was interested in Restinaceae and Central Epidaceae and whether they were the same thing or separate families. And because Trithuria had been put next to Central Epidaceae, she had some DNA analysis done. But the results were disappointing. They were worried that they'd got contamination because the Trithuria didn't turn up when that, so they didn't do anything with it. And then another group. So this is out of place actually. But this is I mentioned that Trithuria is obscure. It's not always obscure. That's a mass flowering Trithuria in a lake down near Lake Utica. I misplaced that slide, apologies. So another group looked at the, I wonder if I've got this one. Yes, this one here. A group of, a multinational group, with principal author being Thysanotus, studied Trithuria with DNA. And they found the same thing as Barbara Briggs' group, that Trithuria wasn't anywhere near where it should have been. They were absolutely astounded. Their paper was published in Nature, which shows the prominence of this discovery. And in recent years, from about the 90s onwards, there's been a great deal of DNA research on the flowering plants, and it's refined our ideas of the relationships, and there's been some fairly major realignments at times, but no realignment is more outstanding than this one. It's an astounding discovery. This might be what you heard about here. Yes. So this is a typical tree diagram of the results of a DNA analysis. And what we find here is that Amberella is a small tree or shrub from New Caledonia that always seems to come out at the base of the flowering plant tree. And also we've got, and this is where Trithuria came out, in this group, the next group, next lowest group on the flowering plant tree, along with things like the water lilies. So these other things are all water lily type plants. And then there's a bunch of other unusual things, mostly from tropical rainforests. in that next group. And then there's another group with some other unusual things that just don't go into the normal classification of the monocot dicot system. So what this means is that the monocots are here, quite high up in the tree, and in amongst dicot groups. And this is the first group that are regarded as proper dicots. So these ones are all collectively called basal angiosperms, not dicots or monocots. I'll explain all this in there in a moment. Thysanotus moved from here all the way down to here when in the classification when DNA was used. Returning to this slide, I'm sure that most of you know the differences between monocots and dicots. So for a very long time, flowering plants have been classified in those two groups, and it's based on whether they've got a single cotyledon or two cotyledons, and there's a suite of other things that generally correlate with that. Did I mention that Trithuria, with its simple flowers, doesn't have any petals, so we can't use the number of petals criterion, and some others wouldn't apply either. But generally speaking, it fitted into the monocot group. But now with the DNA studies that we've got, monocots and dicots are not accepted as simple two groups of flowering plants. Monocots are still accepted, but there are several groups of dicots. It's still a reasonable thing to talk about, though. So the Trithuria was in a group high up, as we would say, in the tree of the monocots, among the poales, the grass group, green pollinated plants that black and colourful petals as they don't need it. And they were always considered monocots. And I need to say a little more about that. There were 2 or 3 studies that were looking at Trithuria in relation to its cotyledons, and it's been generally considered that Trithuria had one cotyledon. That's one of the reasons why it was regarded as a monocot. It had all those other features that seemed to correlate with these things here. But people had looked at various stages. They didn't always have access to all the stages of the developing flower. And there's a botanist in the United States called Ned Friedman who acquired some seeds from a colleague in Victoria. So seeds from swampy Victoria sent to him. He grew them, and it's quite an achievement to grow those things, but he did. And that gave him access to all the developing stages of the flowers, and his specialty is looking at the development of seeds and the embryology of plants. And he's a marvellous anatomist. He does beautiful microscope slides, and so he's able to get good information. And with Trithuria, he found that there are actually 2 cotyledons. One of them is very much reduced. That's the reason why people have replaced it. But there's an interesting story all on its own about that. So that our interpretation of that would be that the earliest flowering plants had 2 cotyledons, and that later on some of them, the ones we call monocots, lost one and became one cotyledon. And Trithuria evolved before that happened, so it's got 2. So this is perhaps a slightly friendlier, more modern diagram of the relationships. I'll just put it in because they have a bit more colour. And this one here, Liliani, these are the gymnosperms, the pines and so forth that are not flowering plants. And down here we've got the Nymphaeales, which Trithuria sits in. And these are its relatives. The opposite, which is small and obscure. Big, showy, colourful petals, lots of them. Still liking water though. So that's a very interesting thing. And what that means is that if the basal angiosperms contain big showy flowers and small simple flowers, it isn't a help for working out what the first flowering plants were like. So that's still a question that's been looked into. So this is where I want to talk about the importance, the scientific importance part of it comes into. As it's considered one of the earlier evolved flowering plants, it means that Trithuria really needs to be included in any study that's looking at questions of what the earliest flowering plants look like. And it's regularly used for that. And as soon as that Nature paper came out, there was an explosion of research on Trithuria from Australasian, which is how I got involved with the various overseas people. One of the reasons why I was involved was that there were more species here that are quite accessible, and I knew where they were and how to identify them, so that made me valuable as well. And quite a number of scientists have come to WA to see or collect Trithuria, including a couple of PhD students use this group as a basis. One student, Mackenzie Taylor, was from Tennessee in the United States, and she spent 2 seasons based around Manjimup, where I was working at the time, out in the swamps, fighting off mozzies and doing minute manipulations on the flowers. It was very successful. So they're obscure now, but famous. But as I showed with that picture of the nest flower, they're not always obscure.